Greetings, and thank you for the opportunity to present at Writing the Future of Cancer Research. Today, I will provide a brief presentation that describes the capabilities of the Twist methylation detection system in detecting cancer biomarkers using critical, clinically relevant sample types, such as cell-free DNA. In order to help scientists and researchers advance the field of cancer research, Twist has worked tirelessly to develop an advanced end-to-end targeted methylation sequencing workflow. The system is a step forward in targeted methyl-seq, providing best-in-class sequencing uniformity and sensitivity. We accomplish this performance through three key advances. 1. State-of-the-art enzymatic conversion that replaces classic bisulfite approaches. 2. Advanced probe design techniques to enable efficient post-conversion capture. And 3. Optimized fast hybridization conditions that ensure uniformity and saves days in samples to sequencer processing. The end-to-end workflow includes ligation-based library preparation and enzymatic conversion reagents in the NEBnext EM-seq for Twist targeted sequencing kit. The library preparation and conversion have been specially optimized for use with downstream target enrichment. This is coupled with a modified fast hybridization workflow that includes a key methylation enhancer component to ensure maximum on target for efficient sequencing. Twist has optimized an advanced design algorithm to deliver the final piece of the workflow, the custom methylation panels. Over the next few minutes, I will describe how these three key components come together in an optimized workflow to deliver a best-in-class solution for targeted methylation sequencing to better understand the underpinnings of cancer from challenging samples like cell-free DNA. The library preparation and conversion module features the NEBnext EM-seq for Twist targeted methylation sequencing kit. In order to detect the presence of methylation within the genome, conversion of cytosines is performed. Unmethylated cytosines are converted to uracil that is read as a thymidine residue in the final sequencing data. Cytosines that are methylated, however, are not converted, and the presence of C in the final sequencing data is then used to identify the sites of methylation. A common technique that is used for conversion is the use of chemical bisulfite conversion. While this is effective at converting cytosines, it has unfortunate side effects of damaging the DNA, leading to compromised data quality, expensive sequencing, and limited sensitivity. Twist has partnered with New England Biolabs to provide an enzymatic approach in this workflow that eliminates the damaging bisulfite treatment step and greatly improves sequencing metrics. The improvement in sequencing compared to bisulfite approaches is highlighted by global examination of GC representation in targeted sequencing data. Across the targeted regions, the enzymatic method in the EM-seq kit shows more even coverage, with a significant improvement in representation in regions of high GC content, which is particularly relevant for studying methylation that most frequently occurs in the context of C followed by a G in the genome. These CpG motifs are densely packed in regulatory regions of the genome that drive gene expression, precisely the regions of interest to cancer researchers. The second piece of the system that ensures outstanding performance is the Twist custom panel design. As mentioned, methylation sequencing and capture involves conversion of unmethylated cytosines to uracils while leaving methylated cytosines intact. By performing the enzymatic conversion prior to capture, a variety of different species are generated and fed into the target capture. Twist has implemented an advanced algorithm to ensure that there is capture of all the species present. This includes sense and antisense, as well as methylated and unmethylated sequences. Through extensive research and machine learning, the design is adapted to the custom regions of the genome and can be fine-tuned for stringency to meet the needs of a particular experiment. Here we see the results of the advanced design algorithm. In these experiments, samples with different levels of methylation were converted, enriched, and sequenced using the workflow described. The data demonstrates the consistency of the hybridization conditions to detect content in the range of 5% to 100% methylation. Furthermore, The differentially methylated regions, or DMRs, are detected consistently across a range of 25% to 100% methylation, indicating efficient capture of a broad range of methylated species in the samples. The third key advance within the workflow is the hybridization. The Twist fast hybridization workflow has been specifically optimized to ensure consistent performance with an efficient workflow. With a hybridization time of less than 4 hours, this can save days from a typical targeted methylseq workflow. Further, the use of methylation enhancer helps to ensure maximized on-target rates, resulting in more efficient sequencing that can lead to better data quality and cost savings. The optimized hybridization conditions lead to efficient capture across a broad range of panel sizes. Here we see performance metrics for 3 designs ranging from 500 kilobases to 50 megabases in size. The performance across this broad range is outstanding, with high on-target rates, even coverage as demonstrated by a low fold 80 base penalty value, good depth of sequencing, and efficient use of data as shown by the low duplication rates. Now that we have an understanding of the system's performance, we can see how this is applied to more clinically relevant sample types, here with cell-free DNA. The use of cell-free DNA, often derived from plasma samples, offers a unique and less invasive way to assay a patient's DNA for cancer markers. One important marker that is emerging is patterns of methylation in the human genome that can be used to track the onset, progress, and regression of cancer. The quantity of cell-free DNA is often quite low, and this can pose significant challenges in detecting changes in methylation patterns in DNA from normal cells compared to that of cancerous cells. This is even more challenging when using chemical bisulfite conversion that damages the DNA. Here, we see the significant advantage of using more gentle enzymatic method for conversion. Excellent conversion rates are coupled with less damage to the DNA, leading to higher library yields. Another way to examine the data to see the difference between chemical bisulfite conversion and enzymatic conversion is to examine the fragmentation patterns often seen in cell-free DNA. The DNA that is present in plasma can remain more protected from degradation when wrapped around histones. Because the histones are a fixed size, the DNA bound to it is often seen in distinct fragment lengths of approximately 150 base pair, roughly the number of bases that wrap around a single histone octamer. The chemical bisulfite treatment, seen on the left, tends to cause breaks between histones that are connected by DNA, leading to a single, uniform fragment size. The more gentle enzymatic fragmentation, on the other hand, preserves a much higher proportion of these longer DNA fragments, leading to more valuable information about the regions of the genome that are methylated. The enzymatic approach also provides for detection of more methylation target sites in the genome, without introducing detectable bias in the sites detected. In cell-free DNA samples, the enzymatic approach detected an average of 15% more CpG methylation sites. For researchers using cell-free DNA, this means the Twist targeted sequencing approach can enable better detection of more potential biomarker sites in these critical sample types. In comparison of cancer samples to normal samples, key differentially methylated regions of interest were clearly identified. It is these regions that are the most critical for connecting the methylation patterns observed in targeted methylseq data to the progress of the disease. The differentially methylated regions are an attractive biomarker for detection because of the numerous sites that can be detected. This contrasts with a single point mutation, as an example, in standard approaches that can be much more difficult to measure consistently. The Twist methylation detection workflow is the most advanced set of technologies to enable cancer research, leveraging state-of-the-art enzymatic conversion, advanced probe design algorithms, and a fully optimized hybridization workflow. These all contribute to the best data in the most cost-effective way and can be adapted to a broad range of different target sizes and genomic regions. In closing, I want to thank you for the opportunity to discuss the impact of targeted methylation sequencing on cancer research.